We are live now.
We are live now. Guys, अभी तुम लोग को म्यूट होते रहना पड़ेगा। अगर अभी सिर्फ अमोराज मैम और जो प्रोफेसर आएंगे वही बात करेंगे। ओके, सो लेट इट गो। अभी मैम आएगी। अभी कुछ इश्यू आया तो मुझे जल्दी से मैसेज कर देना। रुशाद, hello ma'am. I am Riddhesh. I am secretary of Bombay Technologist, and Rushad is also present in this meeting. Thank you for this lecture. And Amograj would be the host for today.
Good evening, ma'am. I am Amograj from Bombay Technologist. We are looking forward to host you today, ma'am.
Hello.
Yes, ma'am. You're audible.
Can you? Okay, fine. That's what I was.
Yes,
So
ma'am.
should I share my PPT now? Just check in case if
No.
there is any.
Yeah, ma'am. You can check that. You can check that.
Okay, I'll just share it. Can you see it?
Yes, ma'am.
Okay, good. So I'll wait for another 10 minutes, right?
Yes, ma'am.
Okay.
We'll be starting in 2 minutes exactly.
Okay. Sure. So one thing I have to ask, should I keep my video on or is it fine if I keep it off?
Anyway, as you are comfortable, ma'am.
Okay,
Whatever is
great.
comfortable is fine, ma'am.
Okay, I will keep it off then. Okay.
Excuse me, ma'am.
Yeah.
Ma'am, it would be great if you could speak a bit louder. Your
Okay.
voice is a bit.
Okay.
Okay, ma'am.
Now, is it okay? Hello?
Yes, ma'am.
Okay, great.
So, good evening, everyone. Welcome to the first BT talk of Bombay Technologist. The Bombay Technologist is an in-house student-run, peer-reviewed journal of the Institute of Chemical Technology, Mumbai. We have a legacy of 7 decades with 66 volumes of journals published. Bombay Technologist journals can be read in various libraries across the world. We welcome all our viewers for this amazing talk. We have with us today our guest of honor and eminent speaker, Dr. Shilpee Jain, for the session Smart Magnetic Health Systems. Ma'am has been working in the field of biomaterials for various therapeutic applications for more than a decade. Dr. Jain did her PhD from Indian Institute of Technology Kanpur in 2013. She has received Inspire Faculty Award and IUSSTF Fellowship to work in the United States. Ma'am has also been developing multifunctional micro devices for various biomedical applications with electric and magnetic simulation. After working in academy as an Inspire Faculty Fellow at the Indian Institute of Science for 5 years now, she is now proceeding to launch a startup to translate her research outcome from bench to bedside. With immense pleasure, we invite ma'am for her presentation. Over to you, ma'am.
Yeah, thank you everyone for organizing this talk, and especially for Ritesh and Rusha. They have been in contact for few days to organizing, arranging everything smoothly. Um, so today I'm going to talk about smart magnetic nanosystems, something about our research work, how we have started, and where we are currently. So, first of all, I would like to give a brief idea about magnetic materials because I'm not going to give a detailed um, uh, structures related stuff of magnetic materials. So, as you all might know, the magnetic materials broadly divided into three categories: uh, diamagnetic, paramagnetic, and ferromagnetic materials. So diamagnetic materials also, you can say those are non-magnetic materials, for example, gold. So if you keep gold near to any permanent magnet, you see it will always uh, repel. Whereas ferromagnetic or paramagnetic materials, for example, liquid, it is paramagnetic material. It's a weak magnetic material. So when you place liquid nitrogen near the permanent magnet, then you will see it will attract. But as soon as you remove these magnets, this liquid nitrogen becomes non-magnetic. So they are magnetic but very weakly magnetic, and they have the magnetization value very in a lower scale. Whereas ferromagnetic materials, they are very strong magnetic materials, and when you place these materials near the permanent magnet, then they will attract to it, and I will show in my next slides how these materials, their unique properties will you can use for various therapeutic applications. So in whole my talk, I will focus mainly on ferromagnetic materials, that is strong magnetic materials, for various therapeutic applications in the form of bulk as well as nano particles. So these magnetic materials are
Excuse me,
having,
sorry to
yeah,
interrupt you, ma'am.
sure.
Ma'am, could you please increase the volume? The
Okay,
voice is still low.
okay. Now, okay,
A
I
bit
think I have
more.
to maybe the my mic. I hope it is okay now.
Yeah, ma'am. It would be great if you could increase your volume a bit
Yeah,
more.
I've already yeah, increased it.
Okay, ma'am. Thank you.
Okay, yeah. Please interrupt me if there is any problem in my voice or anything. Okay.
Sure, ma'am. Sure.
Okay, so now come to the unique properties. So first unique properties I would like to talk about the residual magnetization. So you must have seen this with any strong magnets, magnetic materials such as iron oxide or iron nails, steel nails, that if you magnetize it or just rub permanent magnet on any nail, then it will become magnetizer. Even if you remove that magnet, it will still be magnetized for some few minutes, and you can attract other materials using that magnetized nail. So this phenomena is called residual magnetization. And the other interesting properties of magnetic material is wireless movement. I'm sure during your childhood you must have tried using two magnets just moving around on the paper, and it is really fun. So here also these iron particles just spread on the top of the paper, and you can move them without touching, without any contact, using some magnets. These are two very important aspect or very important properties of magnetic materials, which none of other materials show these properties. And using such those properties, we are advancing our healthcare system. So why, I will first explain why these magnetic materials, strong magnetic materials, show residual magnetization. I'm sure some of you already know about the phenomenon behind it, but I would like to focus it. I will just briefly tell you what is the reason behind it. So, the strong magnetic material in the form of bulk or bigger size, they, they are made up of small, tiny magnets, that those are called small domains. In a single domain, all the magnets, tiny magnets you can say, the magnetic moments are aligned in a particular direction. So there are so many tiny domains in which magnetic moments align in a particular direction, where, and if you see overall, all the domains have random magnetic moment. So when you apply magnetic field, then all these magnetic moment try to align in a particular direction. So this is called the magnetization. So when you increase the magnetic or keep applying the magnetic field, then the magnetization or these magnetic moment or in all the domains, they will try to move and align to this in the direction of the external magnetic field. At some point, these all the dipole moments, they aligned in a direction of magnetic field. So when you further increase the magnetic field, there will be no change in magnetization, and this point is known as the saturation magnetization. So, but when you remove the magnetic field, these materials are not able to reverse their direction. So some of these dipole still show, still aligns in the direction of magnetic, external magnetic field. And in order to reverse their direction, we have to apply the magnetic field in the reverse direction. So this is actually kind of external or extra energy you are providing to change the direction of this magnetic material orientation, the dipole orientation. So this whole thing is called as a hysteresis loop. And so as you know, this energy, extra energy you are providing, it is not just disappeared anywhere. This extra energy you are providing is converting into heat. It's kind of loss, and the heat loss is only depending on that area under the curve. So basically that is the property of the material. Some material shows smaller area of this hysteresis loss, that means they are losing less energy or producing less heat compared to the other materials. So but and this energy, basically this heat loss we are using here to provide heat or increase the localized temperature of tumor site at the tumor site to treat them. But the interesting part here, when you decrease the size of this material, the number of these domains are also reduced. And at some point, at the nano scale, there will be only single domain. So as you can see here, there will be no hysteresis loss, there will be no coercivity, only saturation magnetization is high. So this single line you can see here, this represent this behavior is similar to paramagnetic material, but as this saturation magnetization value is very high, we referred it as a super paramagnetic materials. In this case, in these nanomaterials, this still shows they still generate heat when you apply AC magnetic field, but not because of hysteresis loss, but because of nil relaxation or Brownian motion. Then Brownian motion, because these particles are very small in size and they are not stable at room temperature, and they keep moving. And because of external magnetic field, when you apply magnetic external magnetic field, these domains, the single domain, the magnetic dipole moment also keep changing. And these two phenomena uh, just help to increase the surrounding temperature. So it is not necessarily we have to use bulk material or bigger particles to generate heat. We can use nanomaterials to generate heat and increase the surrounding temperature. So now I will come to hyperthermia, how this material used to enhance the temperature and used for the therapeutic purpose. So this therapy to increase the surrounding temperature near 42 to 46 degrees Celsius is called the hyperthermia therapy. So you can provide heat to um, relieve your mild pain. At home, usually we use hot water bottle to just remove the relieve our mild pain. And at little higher temperature, even we can use this temperature, use heat to kill unwanted cells. So recently, someone asked me this question that in North India, a lot of people uh, are just going out in summer because, and there are the temperature is around 48 49 degrees Celsius. So someone if having cancer, and it would be possible to treat that cancer if that they are standing in the sun. So but my answer is of course no, because it's not possible. Because in heat, when you come out in the sun, that heat try to increase the temperature of your body, but your body have some feature, it can maintain your temperature normally in a healthy range by sweating, and it will work to dissipate the heat because of your environment, and it maintains your normal healthy temperature. So what about the other small stuff aids you use at home to increase surrounding temperature? For example, water bottle, hot water bottle, and heating pad. Those are also can provide some heat, but still you can't control that amount of heat or the localized. I think the penetration is not very good enough to kill the cancerous cells. So, what are the key factors which decides to use any or design any system to provide localized heat which can penetrate inside tumor cells and kill those cells. So the most of the first important factor is basically is the material, type of material, as I already discussed, that hysteresis loss or maybe some other phenomena, nail relaxation and Brownian motion, those are dependent on the type of material. And size and shape also, this both shape and size affects dramatically in the production of heat. And of course the external magnetic field strength, the frequency, and the magnetic field intensity, these two factors also decides the dissipation of heat from the material. And the second stuff, it also depends on the organ where you are using or applying this heat. For example, very compact or internal tissue organ, or because various cells, they show different behavior when they are exposed to heat. So it depends on the type of tissue, it depends on the blood flow and the thermal conductivity of the tissue. The various cells show various different range of thermal conductivity. So it's not necessarily that one system you are, or one heating parameter you are using to some particular kind of cells, that those same parameters you can use to kill different kind of cells. So we had to optimize all the parameters specifically for particular disease or particular type of cell or tissue. What are the other available, other than these magnetic materials? There are some other ways to provide localized heat. Uh, The first one is the radio frequency ablation. Ablation is also form of hyperthermia. It's but at the higher temperature range. So hyperthermia, you can say in the temperature in the range of 40 to 46 degrees Celsius. Whereas if you apply temperature above 48 degrees Celsius, then this process is known as ablation. So in the radio frequency ablation, you have to insert a needle which will provide, uh, by which you can send high electrical power to increase the surrounding temperature. So this is not fully non-invasive, but still it's not very effective, because there are a lot of other problem associated with this therapy. And other therapy also, ultrasonic, ultrasound hyperthermia and photothermal hyperthermia. These, all these three are still at the lab scale, and there are not even a single clinical evidence where people have used these therapies successfully. But a combination with other therapies, for example, in the case of radiofrequency ablation, with some chemotherapy or chemo drug, or after the surgery or in combination of surgery, these therapies are somewhat effective to increase the efficiency and reduce the recurrence of the cancer. So now come to the magnetic hyperthermia therapy. Magnetic hyperthermia therapy is non-invasive therapy, and the system. So I first talk about the system you decide to, any kind of system to decide to make, to use for this to provide heat to the tumor tissue. So first we have always have to consider the biocompatibility. It should be biocompatible, even though if you are not implanting any material inside the body, even if it is interacting for a short period of time to your body, but still it has to be compatible to your system, because it will definitely come to your directly contact to your healthy tissues as well. So, and the other factor is the stability. Your system should be stable if it is coming in contact with your body fluid, because body fluids are are very corrosive in nature. So if your systems are not stable, and if your material is only in interacting for a few days, it can still generate some toxic agents to affect your healthy cells. So these are main parameters to design any material to provide heat to your body to kill the cancer. So there are literature where people are using suspended nanoparticles. Those are called ferrofluids. Those are iron nanoparticles. They show very good heating property, and you have to just inject those suspended iron oxide particles at the site of tumor, and you can apply a alternating magnetic field to increase the surrounding temperature. But there are certain challenges which, where because of those challenges, it's really very difficult to use these systems. uh, so the first thing is the agglomeration. It is really difficult to control the agglomeration of magnetic nanoparticles, because they are having high surface energy, and uh, of course because of magnetization, magnetic properties, so they tend to agglomerate. And once they agglomerate, it's difficult to break those agglomeration. And if we use agglomerated nanoparticles to provide heat, then there will be ununiform heating. And because of this ununiform heating, you will not be able to destroy the tumor cells effectively. And the other problem with those using nanoparticles, the cellular uptake, the other healthy cells can also engulf those nanoparticles, and wherever those nanoparticles are present, they always produce heat when you apply external magnetic field. So once these healthy cells will also engulf, and you apply external magnetic field, you can basically destroy, can also kill your healthy cells along with your tumor cells. So these are some certain issues we have to think about before we use these suspended nanoparticles for therapeutic applications. There are certain ways by which we can overcome such challenges. For example, that agglomeration, we can coat these materials with some polymers, those some biocompatible polymers, or we can um, embed these nanoparticles in some sort of hydrogels. So in our case, instead of using or coating our nanosystems, we try to encapsulate these particles inside the fibrous matrix. So here are some images, SEM images we use to provide heat or use these systems to generate heat to treat the cancer. So first, briefly, I will tell how we have designed or produced these fibrous matrix. We used electrospinning technique. In this technique, we use a mixture of PCl, polycaprolactone, and iron oxide, suspended iron oxide nanoparticles, and we play with various solvents. And as you can see here, we were able to get various structures. Uh, for example, this smooth structure in which the nanoparticles are embedded, and you can't really see from that top of top or externally that these particles are in the fibers, whereas in the other type of structure, in the porous fibers, these particles, if you can see, they are sitting somewhere in these pores. And this rough kind of fibrous structure, if you see closely, all the nanoparticles are sitting on the top of this fiber, and this structure is very interesting because compared to all other smooth and porous structures, these fibers structure, you can see here, they are generating higher amount of heat or increasing surrounding temperature when you place inside the magnetic field. So, and this is the broad, it is, if you see these fibers, it's like normal tissue paper, and it's very flexible, so you can easily cut into any shape and size, and you can use for your application, different applications. Well, the other interesting factor about this nanofibrous mats, we also incorporated drug in it, and these fibers easily can diffuse drug while we, even in the absence of heat or in the presence of heat. So here, as you can see, when we don't apply any external power, these fibers diffuse drug in a small amount, but when we apply this the external AC magnetic field, these fibers loses its texture and release the almost 90% of contained drug. So, and but the other important thing you can notice here, these magnetic nanoparticles, they are firmly attached with all these polymer matrix, and they are not losing it. So we can make sure that these nanoparticles are not releasing from this nanofibrous matrix even after applying heat, and even, and also they have changed or completely deformed after and the use of magnetic field. And so we can make sure that there is no side effects of or toxicity of this system if we use for any clinical purpose because of that accumulation of the nanoparticles. So before doing any animal testing or using for uh, to treat any cancer, we had to test this system in vitro using few cellular model to whether they are working effectively or not. So first we tested our system using HeLa cells. These are cancerous cells. So if you see here, this green part is the stained cytoskeleton of the cell, and the blue is the nucleus. So we seeded cells, and we just suspended our fibrous matrix with a specific weight and shape and size, and we placed this whole tube in the magnetic field. So within 10 minutes, we were able to see the cells completely lost their shape and size, and ultimately they dead. So no matter what kind of fibers you are using, we are able to achieve or increase the surrounding temperature by 10 degree in 10 minutes. And we can see, you can see here that all the cells are around 70% of cells are dead only in the presence of heat. So here, if you see those diagrams, the uh, lower half is showing the live cells, and upper half is showing the dead cells, number of dead cells. So here, when we use drug and heat uh, using those fibers, that you can notice here almost 90% of cells are dead within 10 minutes of time. So at this point, this number of dead cells is the point of no return, means that cells can't revive again once you remove this heating or alternating magnetic field. So at In vitro, we found these systems are very effective. You can kill almost all the cells within 10 minute time duration. So what are the reasons why this heat is so effective and how it kills the cell? So there are a lot of parameters, as I mentioned previously, that external magnetic field strength and type of material and all those big parameters decide your heat. But at the cellular scale also, as I mentioned, that the type of cells, their conductivity basically plays a significant role to decide whether cells will die or not. So the temperature range um, also is the other parameter uh, to to decide what whether cell will follow apoptotic or necrotic cellular death after this heat exposure. For example, if we use um, magnetic field strength or material in such a way to generate only uh, temperature, let's say 40 to 43 degrees Celsius for short period of time, then cells will not uh, uh, immediately kill. They will follow the apoptotic pathways, and maybe they will take 24 hours to finally follow the societal attempt. So this apoptotic cellular death is called uh, cell suicide. So they follow the particular pathways, and then ultimately cell will die. Whereas if we use a higher temperature, the cells will go now necrotic cell death. This necrotic cellular death means that nucleus and other cellular organelles, they will immediately destroy, and within few hours you kill your cells. So the other parameters of cellular mm, rigidity and shape also comes in picture when you apply temperature in the lower range. For example, that the cell will initially change its shape, and there will be no protein synthesis, and protein will also denaturation, and protein will agglomerate near the nuclear matrix, and there will be no formation of RNA and DNA. So these all things will take time. If we are uh, providing temperature in a little lower range and for a shorter period of time, then cell will take their time and they will die in a like 24 hours or maybe longer. But if you provide little higher range of temperature, then you can kill your cells immediately without even uh, inducing or starting all these other phenomena. The other interesting feature I can share with you that this hyperthermia in the lower range can also reverse the drug resistivity. This is the drug tolerance behavior, or the chemo drug resistant behavior of the tumor cells is very very problematic when we treat the cancer. So in the chemotherapy, when you use or when you provide doses of chemo drugs to treat cancer, these chemo drug basically uh, induce apoptosis in the cells. But at certain point of time, these cells becomes very tolerant, or they produce such kind of gene expression by which they become drug insensitive. So this problem with chemo drug resistant, you can't further use that therapy to treat your disease. But if you apply heat in the lower range, you can reverse this drug sensitivity or resistivity in the cells. This lower range of temperature or short pulses, short doses of hyperthermia can reduce those particular gene. Those are responsible for drug sensitivity, and ultimately your cells become resistant, and your cells become sensitive against to your chemo drugs. So there are two things when you when you use uh, chemo drugs or drugs in the presence of heat in order to enhance your efficacy of the therapy. So the first thing is the heat is reversing your um, drug resistivity, or that's increasing the sensitivity towards the drug. And the other thing, the heat also enhancing the activity of drug. So these two factors merging together, and with the heat you will be able to achieve that higher amount of cellular death in a short period of time, and very effectively you can kill cells basically in a short period of time. So you can basically enhance efficacy of your system, of your therapy, by using this combined therapy. So now I will discuss little more about the particular cancer, skin cancer, for we use our system to treat. The fibrous matrix uh, uh, we fabricated in our lab. So we got very interested, and we thought we should use for certain application. And the skin cancer is always on the outside of your exposed externally. And in the current form of our system, it's difficult to use for other kind of cancer, because if you treat other kind of cancer, you have to open up that area and place those systems. So it will not be very it will not be a non-invasive technique anymore. So whereas for skin cancer, we can use in the current form. We made bandages in the form of bandages, and uh, we use external magnetic field. But before going into detail how we have treated the tumor, induced tumor in the mouse, using such as those bandages, first I would like to discuss that type of skin cancer, and what are the problems associated with the current therapies available to treat this skin cancer. So there are two broad categories of skin cancer: the non-melanoma skin cancer and the melanoma skin cancer. So non-melanoma skin cancer is the most common in the USA. 1 in 5 people are affecting or getting non-melanoma skin cancer. Even in India, also people are working in the farms. Those are really very uh, kind of getting this kind of cancer. And melanoma skin cancer is it's not very common, but still it's a deadliest cancer. A lot of people are dying because of this melanoma skin cancer. And this category is basically depends on the cells which are producing or producing those cancer, the location of the cells. So currently there are a lot of therapies available to treat such skin cancer. First is the surgery. That is the first potential therapy to completely remove the cancer is the just cut that area or remove those unwanted or affected cells. But here is the problem: if the size of tumor is more than 2 centimeter, or if someone is having more than 3 or 4 tumors, then it would be difficult to treat or remove all the tumors using surgery. That is the difficulty with the surgery. And the other problem using certain patients are also you can't really use surgery. For example, um, if patient having that diabetes or certain other medical condition, that you can't really operate on them, so you can't use the surgery or more graphic surgery that to remove the cancer. So in that case, the radiation therapy usually used to remove that unwanted or cancerous cells. But the radiation therapy is not really very um, promising because it the radiation itself can produce that second type of cancer if you expose. In the skin cancer, the main reason is the UV exposure, that's radiation exposure. So in the radiation also, it can also generate, if you treat the one kind of cancer, it can also there is a risk to produce certain other uh, relevant cancers. So it's very limited use, and some one person cannot have, there are fixed doses, so one person cannot have beyond a certain doses of radiation in their lifetime. So there are lot of constraints to use these therapies to treat cancers. And of course, that chemotherapy and photodynamic therapies are also available to treat those melanoma and non-melanoma skin cancer. But as I mentioned previously, chemotherapy, again, the cells become resistant, and those are not effective. And the photodynamic, there are a lot of chances of recurrence of cancer. And again, if tumor size is more than 2 centimeter, then this therapy is difficult to use. And the photodynamic therapy is also very limited in clinical trials because it's still at the early stage. So in our case, we use those bandages as a potential therapy for the treatment of skin cancer. In this, we use a mouse as a model to test the efficacy of those systems, whether and to optimize all the parameters uh, to the to get the best external magnetic field range and the size and the amount of magnetic fibers made to generate enough amount of heat at the site of tumor. We in for this test, we induced tumor on the mouse skin, and we gave. So the other point, other thing also I would like to focus here, the external magnetic field strength also. It's not possible to apply very high frequency beyond certain range, because there is a certain threshold limit we have to consider, which is which a human being can tolerate. So there is, so we can't apply like 500 kilohertz and a few Tesla altogether to generate heat. We can no doubt get achieve temperature within a seconds, but still that will be harmful to our body. So we have to keep our system in a healthy range, and without affecting our nearby healthy cells, and only effectively killing only those uh, diseased cells. So we were able to achieve all those uh, parameters using those systems. We are able to um, deliver heat localized without in the range of human tolerance limit, and without affecting the healthy cell. That is the main main factor to decide whether we should go ahead with the current therapy or that experimental therapy for the preclinical or clinical trials for the real life treatment or uses. So here you can see after the treatment of the tumor using those bandages in the presence of AC magnetic field, within a month there were we were we just see no we didn't see actually any sign of cancer. So what we did, we placed those bandages on top of the tumor and placed that whole mouse uh, uh, in the AC magnetic field coils, you can see. And so these coils are very, if you touch them, you will not get any kind of shock. Only material which can which are magnetic in nature, only that material will produce heat. So the other part will not affect to this magnetic field. We have tested even without absence of any mandate. We have used similar parameters to see whether this magnetic field inducing or creating any issues with the to affect the normal cells. So we didn't notice any effect of magnetic field in itself. So finally, we after optimizing all the parameters in the range of 236 kilohertz frequency and the magnetic field, it's 3.6 kiloampere per meter. This is very small. It's like it's only around 40 gauss. Um, so during the treatment, we noticed that even I think after 3 sessions of this heating treatment for alternate day. So yeah, here the doses also are very important when you decide whether this therapy, I mean, when you consider the parameters, optimization parameters to for the efficacy. So the doses. So we given these 3 sessions for consecutive days, and then we gave rest for 1 day and alternate days for 2 more sessions we provide. So five in total session we have given for 15 minutes. In that 15-minute sessions we achieved or maintained 45-degree surrounding temperature. We measured the temperature by infrared thermometer, and we also during the experiment we also keep track the body temperature of the animal, and we didn't notice any change in the temperature because that heat is very localized. So, and it was not allowed to increase that body temperature. After these 5 sessions of the treatment, we leave that animal for uh, auto recovery, and we noticed after 2 weeks of this treatment, the mouse recovered completely. Even you can see the hair also um, grow normally, and there was no difference in any kind of activity or weight loss or anything, the treated mouse when you compare to the control mouse without any treatment or without any tumor. So finally, we thought this tumor maybe only top it is treating, but maybe there are something happening inside, and which could be the reason of recurrence of the cancer. In order to check whether the cancer there are any unhealthy or still cancerous cells are present, so we checked this underneath of cells, and we didn't see any notice and I mean, no, not a single cell was present after the treatment, after 1 month of the treatment in a treated tumor area. So the chances of recurrence of cancer, we can say, is very less if we provide the temperature range in the lower range, and for more sessions. For example, if we give high temperature range above 48-49 degrees Celsius, then cells will die effectively and in a short period of time. But the normal healthy cells will also get affected. And there are also, if we give less sessions, then there are chances of recurrence of cancer, because maybe some surrounding cells are not dying because of penetration or proper penetration of heat. So we are making sure here that that penetration and coverage of that tumor area are good enough to just rule out any recurrence of the cancer, and without affecting our healthy cells. So, what are the reason? How this heat itself? We haven't used any drug yet, just heat. We used heat to destroy this tumor tissue. So, what are the reason how these cells are dying? In case of single in vitro test, single cells, there are heat is directly affecting to the cellular level, and cells are following that apoptotic pathways to ultimately or their death. But in the case of tumor tissue, there are a lot of other parameters. There is a blood flow, and there is a lot of compact cells. So if we compare the physiology of tumor tissue with the normal tissue, then you can see that tumor tissue is having lot of a random vasculature. So this is basically the random and compact vasculature to provide the nutrients to uh, unwanted ungrowth cells. So there are a lot of compact cells and vasculature as compared to the normal healthy cells. So normal healthy cells, they are well organized, and their even blood vessels are properly connected to cells. And the overgrowth of the cells in the case of tumor tissue, um, enhancing or producing a lot of compact blood vessels. So this is the basic reason here to kill cancerous cells without affecting the normal healthy tissue when and nutrients. So this is not the direct effect of heat. We can say that indirectly heat is destroying these cells by affecting this vasculature. Of course, if you keep providing heat, then at some point you will directly affect these cells at a cellular level. But broadly, if you see, this is the more effect of the vasculature, destroying vasculature in the case of tumor cells. So, what happens in the healthy cells? A healthy healthy tissue. In the healthy tissue, as it is very open and organized, the heat can easily dissipate and maintain its normal temperature. So you can see here adjacent cells, healthy cells, can maintain their normal temperature, and there will be no effect at temperature of 45-46 degrees Celsius. And because of destroyed blood vessels and lack of nutrition, you can kill your cells effectively. So that was the one thing we had tried at lab scale and in vivo using mouse model, and we are very confident that those systems can also work for other big animals or even clinically. So we are planning to take this forward and use for clinical applications, preclinical trials. The other use of those system I would like to also discuss with you today, that the corneal fungal treatment. This corneal fungal treatment is very difficult to treat currently because the fungal cells, once there is a formation of biofilm, then it's very difficult to treat using any drug, because drug cannot penetrate in the biofilm. So here you can see that eye become completely opaque. There is a kind of film, and if it is left untreated, then there is a chances of completely loss of vision. So in our system, as I mentioned before, that these are very flexible. You can cut in any shape and size. So we have tried to treat, we test it only in at the lab scale, whether we can use those systems to treat this infection. And as for the treatment of cancer, we incorporated chemo drug, but that is not limited in our system. We also tested the release of drug when we incorporated this antifungal amphotericin drug, so that the release of drug from this fiber system similar to those chemo drugs, and the treatment of those fungal cells are very effective. At in vitro we tried, and we were able to kill effectively all the fungal cells um, within 10-15 minutes of time. But still we have to test those system whether that if efficacy, whether they can kill or destroy that biofilm fungal cells in the biofilm. So these are only early stages of those systems. Now I would like to discuss some more other advancement in the nanotechnology. Those the previous fibers matrix, those were big, they were the treatment of external cancers or external infections. But once if we reduce the size, then we can also use those systems to treat deep located tumor using this same heat and drug delivery. So before that, I would like to show you a clip of a movie that is a very old movie, around I think 1960 it released, Fantastic Voyage. So our work is basically inspired from this movie, and I'm sure it won't take much time to take this science fiction into a real life. So in this movie, I'll just briefly tell what happens here. So in this movie, there was a dying patient, and that patient was having some tumor in the brain, and there was no available therapy to treat that tumor. So but the team of doctor, they have certain kind of radiations by which they can shrink the any object. So a team of doctor here, they board a ship, and they exposed to that radiations, and the whole ship basically got shrink, and then they travel to the body of the patient, and then they finally use some laser light to treat that tumor inside the body. So basically here we can't really in real life shrink our human body to travel inside the other human body. So here we basically are making small robots. So those robots can travel inside the body. For example, here they are picking these robots traveling in the blood vessels and picking the dying or deceased white blood cells. So these systems, these robots can go deep inside the body, and they can use or we can just design in such a way to deliver either heat or some specific kind of cells or some kind of drug to treat locally the disease or remove unwanted cells. So basically these systems can use uh, mm, can be a robot, I mean not very complicated machinery robot, but small system which are those are able to travel through the blood vessels. Those are very tiny small devices and can perform a particular task inside the body. But now life at smaller scale changes completely. So how we can design? It's not possible that the similar fibrous matrix I produced and we can just reduce the size and uh, or cut in the nano range, size of nano range, and then use for a specific application. That is not possible because at the nano scale there are a lot of other parameters, a lot of other factors comes in picture. For example, because of the size, because of the small size, the Reynolds numbers uh, reduces and below 1. uh, so I mean what it means. So the human being, if it swims in the water having Reynolds number of the order of 10 to the power 4. So that means a human can easily swim in the water. Whereas fishes, because of their smaller size, they have R value of 10 to the power 2. But microorganisms, um, they are having R value of 10 to the power minus 4. What that means? That means if these microorganisms, if they swim in the water, they travel in the water, it for a human it feels like we are swimming in the honey. So at the lower Reynolds number range, it would be very difficult to move in a viscous environment. It feels like you are swimming in the in very viscous environment. So it's very difficult, no matter how much energy you are applying or pushing forward, backward, but there will be very negligible or displacement or movement. So for this so same thing will happen once we reduce the size of our system. So we need some external power source to move those system or navigate those system inside the body through blood vessels. So there are certain uh, 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 processes available by which we can move those systems, chemically or acoustic propulsion or biological propulsion. But I will just give a brief example that it's not feasible to use all in these different kind of chemical propulsion because there are certain limitations. And for example, in this zinc micromotors, they are uh, they work only in acidic environment. So once they comes in contact with acidic environment, their surface get reduced and they produce hydrogen gas, and this hydrogen gas pushes them forward. But and they can, so there are certain reports available that the navigation of those system inside the tissue. But in those systems there are lot of challenges that we can't control it, their direction. They work only in acidic environment and their lifetime is very short because these systems, once their coating um, the whole coating reduces, and sometime before reach to at the site of the tumor, it get adjusted. So it's not possible to achieve, always not possible, or we can say that efficacy of those systems are not very good enough, because if we send uh, 100 number of systems, and maybe only like 50% or 20% of the systems are reaching at the site of tumor, so it's not very effective. And similarly, in other biological propulsion, we use sperm cells to navigate our system, but that is also not very feasible in real life, because there are chances of rejection or infection. So the best thing is the using magnetic material, because by magnet here, the unique property I talked about in my initial slides, that the control of magnet without any contact, without connecting any wire, is the best way to use here to navigate the system inside the body. So how basically it works? Here is one example. Mm, I don't. Okay, so here you see this is a micro machine made up of magnet, and you can see here it is just moving and taking a sperm cell, and it will just deliver these cells to the egg. And you can easily control the direction of these systems. So when you here we are not using any alternating magnetic field, but to drag the system in the fluidic environment, or in the fluid, you have to use some gradient magnetic field. So once you have that too in a very small range, less than 100 gauss magnetic field, so it will not affect your any other body function. uh, and externally, without connecting even any wire to this system, you can easily, because there is no hindrance of penetration of magnetic field intensity through your body. So basically you are not losing your magnetic field intensity. So in the range, your body kind of opaque to your magnetic field intensity, so you can easily drag it. So when you apply this gradient magnetic field, what happens that a part of this magnetic field will expose some kind of force, and this force will align those nanosystem in a direction of magnetic field, and the torque, this torque will push your system forward. So this is very simple phenomena, like at the bulk we used to do in our childhood, that dragging to magnets, or just moving, navigating, or using other using permanent magnet, the magnetic material or other magnets. So what are the factors? Only the magnetic field intensity here, the gradient magnetic field here basically decides how fast this material will move, but that shape also. Here you can see this spiral shape also pushes, because of other transport phenomena, enhances and move it for this system for faster in presence of lower range of magnetic field. The design is should be very complicated. So the first thing, the design decides how fast those system will move, but it should be the shape and size should also be, we have to consider, we have to think about that where it is moving. For example, if we have to achieve some locations in the heart, then you have to uh, decide its route or its path, where what size of blood vessels it will go through. because we have various diameter of blood vessels, and we really don't want to uh, get stuck our systems in one of those blood vessels, which can inflame, or which can induce other kind of complications. So we want the smooth and controlled movement of those system. For that, we have to consider the size and of course the shape of the system for the faster and effective movement. For example, that a smallest size of the capillary diameter of the capillary in our body that relate connected to heart is 8 micrometer. So our system should be less than or of the order of one maximum 2 micrometer, not in all the dimension. It's not in any of the dimension. If it is more than 8 micrometer, then there is a chances of tilting of this system, and get it can get stuck in that capillary. So those are the factors which are really very important while we design such tiny micro robots for any therapeutic applications. So in our lab we have designed very simple basic feature, not any spiral. First we just produced some rod shape, and and this is not very complicated machinery, but still, since we are able to control its direction in the fluid environment, in the body environment, and they are stable, we call them medical micro robots. um, and the interesting feature about those systems we developed in our lab, um, are they are not only magnetic, but they are conducting as well. So you have two things here. You can uh, navigate those system using external gradient magnetic field, as well as you can use some external electric field also to move those system because of electrophoretic effect. And since the size of these nanoparticles are very small and they are conducting in nature, so the electrophoretic effect is the super electrophoretic effect. Means they are moving very very fast when you apply external magnetic field. So here I'm showing some videos when we apply in the absence the movement of the system in the fluid, in the absence of any external uh, magnetic or electric field, or when we apply uh, um, uh, electric field. Wait a minute, why this. Yeah, so you can see here the movement is very fast in case of electric field as compared to magnetic field because of superphoretic effect. I'm not going into detail. I'll just now come to the next slide, the use of this system. So you can use this system to deliver uh, um, any drug agent or heat, because they contain magnetic material as well. So heat inside the cell, not only outside but inside also, you can provide heat to the cells and for specific uh, disease, to the treatment of the disease. And we have also collaborated with some other colleagues at IISc, and they designed these spiral structures, and those are zinc coated nanomotors, and they were able to move inside the cells, and inside the cells they were able to generate enough heat to kill cells. So those are the things we have tried at nanoscale, as well as a bulk systems to treat cancer or other or other infections for the treatment. So if you see, there are a lot of papers, research articles based on fabrication designing of those micro robots, or systems to navigate, not inside the body but yeah externally, like in my case that bandages. So there are a lot of systems to develop to treat cancer or other infections which are difficult to treat, but still there are very limited clinically uh, uh, clinically there are very limited studies available that they are very good or potentially being using for the treatment. So what are the reasons? Because in a lab scale we are designing the system without considering the problem we can face while we use for real life. So to avoid this, I mean, there is a lack of coordination between biologists and engineers and doctors. We have to coordinate with all these to design a system, because we really don't know what is the requirement to, to treat any disease uh, clinically. Only doctors can tell them what they want, and for that we need to collaborate, and we need to find some kind of bridge between these hurdles, and then only we will be able to make those effective therapeutics in a real life. So all these things I have discussed so far, uh, the systems we can use for various treatments, and using heat or using the drug delivery at the localized site effectively within a short period of time. And I also discussed various size of those systems. You can make those systems to travel inside the body using magnetic field, but these systems are still in that early stage. We have to do a lot of testing, and we have to uh, as I mentioned in my previous slide, we have to discuss with doctors what are the real requirement, what kind of problem we can face while we use these systems for the real life treatments. So it's a long way to go now. Finally, here I would just finish my talk, and I would thank I have advisors, my collaborators, and my students. I was lucky to get really uh, very good students, and of course IISc and DST for all of us. Yeah, thank you.
Thank you, ma'am. Ma'am, before we conclude, we have a few questions in the chat box.
Yeah, sure. So can you please read those questions for me?
Yeah, sure, ma'am.
Yeah. So I don't have to. Yeah.
There is a question from Vandana, ma'am. Is there any particular reason PCL was used for fiber generation? Was any other polymer tried?
Yeah, that's a good question. Actually, the reason why we used BCL, because the melting point, because it has a lower melting point in the range of 60 degrees Celsius. We were able to release whole amount of contained drug. Whereas if we use some other, and the other thing, it is biodegradable. So other biodegradable polymer, we have to optimize the parameters that it should uh, retain for stable during all whole treatment. The PCL is the best. It maintain its in dimension, shapes, everything while we are using for the treatment, and it is releasing whole amount of drug when we use external AC magnetic field, and it is delivering or dissipating heat also. So of course we can use some other polymers as well, but we have to make sure that about that melting point of the polymer and its stability and biocompatibility. Yeah.
Another question from Vandana ma'am. Any stability data on bandage available as per ICH?
ICH. What is ICH?
Uh, she has not made it
Okay.
clear there. She's asking about the stability data, availability of stability data, ma'am, on bandage.
I really don't know what is ICT, but yeah, this
Uh,
availability. Yeah.
I guess ICH is International Council for Harmonization of Technical Requirements for Pharmaceutical Human Use.
Okay. Okay. No. Yeah. So yeah, I am in the process of getting all those certification, but yeah, before that we have to do few more testing for apply for those approvals and all. Yeah.
Uh, Thank you, ma'am. We have a few more questions from our viewers. Uh,
Mhm.
the question states, at higher temperature, protein unfolding and denaturation takes place. The DNA binding proteins also denature, causing chromosomal aberration. How are surrounding normal cells protected from this?
Yes. So, as I mentioned, those effects basically takes place when you directly hitting your cells. But in our case, we are hitting whole tissue. So this is not uh, really, so in case that is the other issue with the nanoparticle. Actually, if you use injected nanoparticles, then you really can't save your healthy cells. If nanoparticles are going inside the cell, then definitely these every phenomena, whatever these cancer cells are following in the presence of heat, healthy cells will also do the same. But in our case, we are providing externally to the tissue, so there is we are harming only that compact vasculature, so it is not affecting directly to that healthy cells. Healthy cells, yeah, because it's maintaining temperature. So uh, there is no effect. I mean, healthy cell can easily maintain, and we are uh, mm, we are not affecting or destroying those. And but in the case of nanoparticles also, we can specifically deliver those tumor cells, specifically deliver those nanoparticles by binding to a specific antigen to um, target cancerous cells, to avoid that destroying of healthy cells by heat and other process by which they can be affected or they can be dead. Yeah.
Okay, ma'am, we have a last question. Does shape of nanoparticle change magnetic behavior of that particle?
Shape of magnetic nanoparticles, yeah, of course. So if you compare the same material with rod shaped nanoparticles and spherical particles, so there are few reports available that that the spherical particles are showing more dissipation of heat as compared to the rod shaped particles. And the other parameters or other factors basically that internalization, that cellular uptake of those nanoparticles, because the cellular uptake of spherical particles are more as compared to that rod shaped nanoparticles. So that heating, ultimately that heating or dissipation of heat effect would be more in case of spherical particles. So those shape also plays a major role to design those kind of systems for heat delivery.
Thank you, ma'am. One last question from our viewer. Uh, it states, does do nanoparticles inside body have immune response?
Yes, there could be, but basically those are biocompatible, so they won't really induce, but of course when we use those systems to kill some internal tissues, to destroy internal tissues, then maybe those debris or that releasing of toxic things may induce some kind of immune response to the body. But yeah, there I mean, as such there is no reported, we haven't tested that, but there are no report available where they cause any kind of problem to the body once they injected or interacted with the body. Yes.
Okay. Once again, thank you, ma'am, for your valuable and informative presentation. And I'm sure all our viewers would definitely go back with a better idea of magnetic systems and their applications in healthcare. We once again express our sincere gratitude for enlightening us on this wonderful topic. I also thank Patravali ma'am for her presence and also all our viewers for their valuable time. Thank you, ma'am.
Thank you for the patience.